Hello, everyone. Today we're going to be discussing an article published in Science Translational Medicine in June 2023 entitled Gut Microbiome Composition May Be an Indicator of Preclinical Alzheimer's Disease. First off, why do we care? Or bottom line up front, Alzheimer's disease is currently ranked as the 7th leading cause of death in the United States, and there's no cure. It's a brain disorder that slowly destroys memory, thinking, and reasoning, eventually leading to the loss of behavioral abilities as well. Estimates of 7 million Americans and 55 million people worldwide, most 65 and older, are living with Alzheimer's. Alzheimer's is characterized by the development of abnormal protein deposits called amyloid plaques and tangled bundles of fibers called tau tangles. On the side here in the first image, you can see the development of the amyloid plaques and the tau tangles in the brain versus a healthy brain. With the buildup of these amyloid plaques and tau tangles, neurons start to lose connections with other neurons. They stop functioning and ultimately die. The damage starts in the hippocampus and the entorhinal cortex, which are essential parts of the brain for forming memories. As more neurons die, other areas of the brain deteriorate and the brain tissue begins to shrink. As you can see in the bottom image, a healthy brain versus severe Alzheimer's brain where the brain tissue has severely shrunk. Next, about Alzheimer's disease and the gut microbiome. The human gut microbiome is a diverse community of microorganisms that contribute to the overall health of an individual. When there is gut microbiome dysbiosis, typically characterized by a loss of diversity, it has been correlated with disease status. In Alzheimer's, it has been thought to progress in 3 stages, from cognitively normal to no clinical signs but with biomarkers of the disease, known as preclinical AD, and lastly, symptomatic or clinical AD. This leads us to the question the researchers of this article are trying to prove, and that is, are there early preclinical biomarkers of AD progression in the gut microbiome? So how was this progression evaluated for the study design? The objective of this cross-sectional observational study was to identify differences in the gut microbiome profiles of healthy participants and participants with preclinical AD, as well as any correlations of gut microbiome features with AD biomarkers, of which 115 participants were healthy and 49 had preclinical AD. Participants were recruited for this study from existing longitudinal cohort studies at Knight Alzheimer's Disease Research Center, or ADRC, in St. Louis from July 2019 to October 2021 in a randomized fashion. Participants ranged from 68 to 94 years of age, and all participants were asymptomatic, with their most recent clinical dimension rating, or CDR, equal to 0. In this study, preclinical AD was defined as CDR equaling 0 and beta amyloid positive. Beta amyloid positive was defined as a value on the centiloid scale of greater than 16.4. The centloid scale is a standardized measurement for amyloid PET imaging. If PET beta amyloid biomarkers were not available, which was what happened in 11 participants, then AB positivity was defined as cerebral spinal fluid of beta amyloid 42 to 40 ratio of less than 0.0673. Healthy was defined as a CDR rating of equal to 0 and beta amyloid negative. Now we will discuss the methodology the researchers used to detect pathogenic beta amyloid and tau proteins, as well as neurodegenerative signs. The first method used was clinical assessment, or the clinical dementia rating scores. The CDR scores were obtained by experienced clinicians. The CDR is used to determine whether dementia is present, and if so, to what stage is its severity. A CDR of 0 indicates that the individual is cognitively normal, whereas a CDR of 0.5 and a CDR of 1 indicate very mild and mild dementia. Next would be OPE status and polygenic risk scores. Apolipoprotein E, or APOE, gene is a well-known gene that influences Alzheimer's risk, and allele 4 increases AD risk. DNA samples were collected at enrollment and genotyped using either an Illumina 610 or OmniSpress chip. Participants either had at least one copy of the APOE E4 allele known as positive or had no copies of the allele known as negative. Next was MRI acquisition. MRI measures were taken of white matter hyperintensities volume, hippocampus volume, and cortical signature. They also used PET imaging to calculate brain beta amyloid plaques on the centloid scale and tau tangles. Cerebral spinal fluid collection was performed via lumbar puncture and analysis for beta amyloid 42 and 40, total tau, and phosphorylated tau 180, all of which are biomarkers for Alzheimer's. Stool collection was collected for bacterial DNA extraction. Food logs and nutritional analysis were performed 24 hours before stool collection. Metagenomic sequencing using NovaSeq 6000. Process reads were used as input to metagenomic phylogenetic analysis computational tool to determine taxonomic abundances, and a unified metabolic analysis software called Human 3.0 was used to functionally profile the metagenomics. And lastly, statistical analysis were performed using students' t-test with Benjamini-Hochberg correction and chi-square tests to evaluate differences in demographics or clinical covariance between healthy and preclinical Alzheimer's groups. Now for the results. In Table 1, it lists the participant demographics at the time of stool sampling stratified by AD preclinical status. On the left, you can see genetic risk data for all participants. Figure 1. Figure 1 shows healthy and preclinical AD individuals that have distinct gut microbiome profiles. In Figure 1A, you can see stacked taxonomic bar plots at genus level stratified by preclinical AD status with color groupings at the phylum level. One thing to note in this figure is that there was no significant difference in Bacteroides to Firmicutes ratio between healthy individuals and individuals with preclinical AD. However, in contrast, Figure 1b shows the principal coordinate analysis, or PCOA, using between sample unifrac distances, which had global differences in gut taxonomic profiles by preclinical status. Global microbiome composition was different between healthy and preclinical AD individuals, after accounting for age, haploprotein E carrier status, diabetes, body mass index, hypertension, and time elapsed between PET imaging or lumbar puncture for beta amyloid quantification and stool collection. Results from Figure 1b prompted a constrained analysis of principal coordinates shown in Figure 1c, which further explains the taxonomic differences in preclinical AD status. Overall, data in Figure 1a, b, and c suggests that the human gut microbiome may change early in AD before cognitive impairment is apparent. Figure 2. Figure 2 shows how gut microbiome profiles correlate with beta amyloid and tau, but not with neurodegeneration. In Figure 2a, you can see pairwise Spearman correlations between microbiome summary metrics in green and AD biomarkers. In blue, you can see beta amyloid, and purple is tau. Orange, neurodegeneration, brown, vascular injury, and in gray, genetic risk factors. Significant correlations are shown with the size of the circle inversely proportional to the p-value. Figure 2b shows linear regressions of Alzheimer's biomarkers against gut microbiome-derived axes. Specifically, regressions of PET beta amyloid, PET tau, or cortical thickness, which is a measure of neurodegeneration, against principal coordinates of analysis, sample coordinates derived from taxonomic profiles in the top and middle rows, or functional pathway profiles in the very bottom row. Overall, Figure 2a and b illustrate that there were no microbiome markers significantly correlated with neurodegeneration markers, and that ultimately neurodegeneration is a later event than the appearance of beta amyloid and tau biomarkers in the brain. Figure 3. Figure 3 shows fitted negative binomial regression models to gut microbiome taxonomic data, identifies species associated with AD preclinical status. In Figure 3a, you have model coefficients on the left and prevalence on the right of top-ranking species significantly associated with healthy or preclinical AD status. In Figure 3b, it illustrates the relative abundances of 10 taxa most associated with preclinical AD on the top row and healthy status on the bottom row. The species most associated with preclinical AD status include the following, and I won't try to pronounce all of those for you, but there were 5 main species, most associated with preclinical AD status. Interestingly, 7 out of 13 of the gut microbial species most associated with healthy status in this cohort belong to the Bacteroides genus. Figure 4. Figure 4 compares the performance of random forest classification models with and without gut microbiome features across combinations of amyloid, tau, neurodegeneration biomarkers, and genetic risk factors for AD. Figure 4a summarizes features included in each of the random forest models reported in figures 4b and 4c. Overall, Figure 4 identifies 7 candidate taxa as important features for classification of healthy versus preclinical AD samples. Feature inclusion is denoted by shaded cells, and models that include or exclude feature selected gut taxa are compared. The last figure is Table 2. Table 2 is improvements in random forest classifier performance after incorporating gut microbiome features. All biomarkers, including beta amyloid, gut microbiome features afforded small but significant improvements in classification, accuracy, and specificity. When PET, beta amyloid, and cerebrospinal fluid beta amyloid 42/40 were omitted, inclusion of selected taxa offered significant improvements in specificity, but not accuracy or sensitivity. Lastly, we will discuss the conclusions of this study. Data suggests that there are about 10 years between when the first beta amyloid plaque shows up in the brain and the first clinical sign of impairment. This means there is a window called preclinical AD, during which biomarkers can be detected and predict disease progression before cognitive impairment has started. Researchers asked whether distinct gut microbiome profiles were present in the preclinical stage of Alzheimer's, and they found that there were differing microbiome compositions and microbiological functional potentials at the preclinical stage of Alzheimer's. Certain microbial pathways were also identified to be most associated with preclinical AD, and they all share succinate as a product. Succinate is a bacterial metabolite produced in the gut that has been linked to obesity, IBD, and now AD pathogenesis due to the gut inflammation that it causes. The pathway most associated with healthy participants produces acetate, which has been observed to inhibit beta amyloid aggregation in vitro and to protect against cognitive impairment in mice. L-stipes, Varicella, and Ortobacter were genera significantly associated with preclinical AD status and had previously been found in symptomatic AD individuals as well. Distinct Bacteroides species were highly associated with preclinical and AD groups, preclinical AD and healthy groups. There was no significant difference in Bacteroides to Firmicutes ratio between healthy individuals and individuals with preclinical AD. Certain bacteria were significantly associated with either preclinical or healthy status, of which F. prausentini Oslobacter species and D. formicigenerans ranked as the three most important taxonomic variables. D. formicigenerans is known to degrade mucin in the gut and play an inflammatory role in the gut. Oslobacter decreases colonic epithelial integrity, and in contrast, F. prausini is anti-inflammatory and was found to be enriched in those with non-AD individuals or healthy individuals. So now some questions to start our discussion. Do you think stool sampling will become a part of the early screening measures taken to detect AD risk? What are your thoughts on the existence of an enteric neuroimmune axis in neurodegenerative disease? Number 3, what further investigations would need to be performed to validate this link? Number 4, this study was only one population group, ADRC. How might results differ if there was a larger, more diverse participant group? And lastly, what are some other limitations that you identified in this study? Lastly are my references. Thank you everyone for listening. Hope you enjoyed it.